It's a pleasure and a privilege to be speaking to you today. You see here my affiliation with the University of Pennsylvania, as well as my disclosures. I'm a co-founder of and advisor to Verve Therapeutics, a company focused on cardiovascular genome editing, which I'll mention in my talk. This is a recent article from my hometown paper, the Philadelphia Inquirer, that featured Avery Watts, who at the time was 10 years old. She has severe familial hypercholesterolemia, which causes her to have very high LDL cholesterol levels. Her disease is so severe that she requires several hours of apheresis every week to clean the LDL cholesterol out of her blood. Despite this aggressive therapy, she suffered bad heart disease at the age of 11 and had to undergo multiple open heart surgeries. This particular article asked the question of whether CRISPR genome editing could provide a new treatment for patients like Avery. The key to treating patients like Avery lies in this woman, Anna Führer, who was featured in the New York Times for a very different reason. She has extremely low cholesterol and extremely low triglyceride levels. She has two defective copies of the ANGPTL3 gene, which means she makes no ANGPTL3 protein at all. Despite this, she's entirely healthy. Imagine if we could transfer the beneficial mutations from this fortunate woman to the unfortunate girl with familial hypercholesterolemia. In principle, it could be done with genome editing. Here's another example where disruption of a gene can have beneficial consequences. About 3% of the general population have loss-of-function mutations in the PCSK9 gene, which is involved in cholesterol regulation. If you have a single loss-of-function PCSK9 mutation, in other words, one broken copy of the gene and one normal copy, you'll have a significantly reduced LDL cholesterol level, as well as a dramatically reduced risk of myocardial infarction, heart attack. There are a few known individuals with two loss-of-function mutations in PCSK9, who effectively make no PCSK9 protein, but nonetheless appear to be healthy. The key point is that people with these mutations do not appear to suffer any harm from having the mutations, only benefit. This has provided a strong rationale for the development of therapies that target PCSK9, with two monoclonal antibodies and an siRNA therapy now available to patients. Traditional therapies like pills or antibody-based biologics have the disadvantage that their beneficial effects only last for a limited time, which means the therapy needs to be taken over and over again for the rest of one's life. The potential advantage of in vivo genome editing therapy is that since it would permanently alter the genome, it could possibly have a lifetime effect and thus might need to be given only once. Although it's not immunological in nature, it'd be similar in concept to vaccination. One very important consideration is the safety of the genome editing therapy. Any off-target editing, that is, editing that causes mutations at locations in the genome besides the target gene, would also be permanent in nature and could potentially have adverse consequences many years later, consequences such as cancer. So we've all heard about CRISPR, but you might not be familiar with the details of how it works. In the interest of time, I won't explain how CRISPR works here. Instead, I'll just say that standard CRISPR genome editing, as shown on the left, is like using a molecular scissors to cut through a gene, with the cellular repair process introducing semi-random mutations that disrupt the gene. Base editing, as shown on the right, is more precise. It's like using a pencil and eraser, replace one base in the DNA with another base, thus introducing a specific mutation, such as a nonsense mutation. Either standard CRISPR genome editing or base editing can be used to turn off a gene. Over the past 8 years, my laboratory at the University of Pennsylvania has been testing CRISPR genome editing and base editing of genes like PCSK9 and ANGPTL3 and other liver genes in various mouse models. In this proof-of-principle study, we used a viral vector to deliver the standard CRISPR genome editing system into the livers of adult mice, where the PCSK9 gene is specifically expressed and produces a protein that's secreted into the bloodstream. The intent of this experiment was to disrupt the PCSK9 gene using CRISPR as a molecular scissors. The question was whether there would be enough disruption of the gene in hepatocytes to cause a sustained reduction of blood cholesterol levels, just as is seen in people who are born with loss-of-function mutations in PCSK9. The answer was a resounding yes. The vast majority of the PCSK9 alleles in the liver were successfully disrupted. This resulted in a 90% decrease in the amount of PCSK9 protein in the bloodstream, as you can see in the left panel. In the right panel, you can see that blood cholesterol levels were reduced by 35 to 40%. This is consistent with what's seen in humans with PCSK9 inhibiting drugs, and you would expect this level of reduction to significantly reduce the risk of myocardial infarction. But because PCSK9 has been altered within the genome, the effect on cholesterol should be permanent. My friend and colleague Keith Jung at Massachusetts General Hospital used the same mouse model to assess the safety of the PCSK9 editing. Using sophisticated techniques, he scanned the entire genome for candidate sites of off-target editing. He then assessed whether there actually was any editing at any of those candidate sites in the livers of the treated mice. He saw editing at only one location in the genome, in the PCSK9 gene at the desired location. This suggested that it's possible to develop genome editing therapies with minimal off-target editing. When we targeted the ANGPTL3 gene in a mouse model of familial hypercholesterolemia using a CRISPR base editor, it yielded even more impressive results, greater than 50% reductions in both cholesterol and triglycerides, a double whammy. The difference can be appreciated by simply looking at plasma samples from treated versus control mice. Now think back to Avery Watts, the young girl with severe familial hypercholesterolemia. Before she undergoes apheresis each week, her plasma looks like that on the left. What if she could receive a single genome editing treatment that turns off the ANGPTL3 gene in her liver and permanently converts her plasma to look like that on the right? It would be utterly transformative for her. Having shown genome editing to be effective in mice, the next step in translation to the clinic was testing in a larger animal model more similar to human beings, non-human primates, in this case, cynomolgus monkeys. Here are the results of work I did with a company I co-founded a few years ago, Verve Therapeutics, published just last month. We used lipid nanoparticles, or LNPs, a technology for which Dr. Daniel Anderson, who spoke earlier in this session, was one of the originators. We developed an LNP formulation encapsulating the RNA components of a CRISPR base editor, as it happens, very similar to the COVID-19 mRNA vaccines from Pfizer-BioNTech and Moderna that hundreds of millions of people around the world have now received. So when I spoke earlier about a vaccination against heart disease, it was a bit more literal than you might have realized. In short-term studies lasting 2 weeks, at different doses of the PCSK9 LNP base editing therapy, we achieved very high levels of PCSK9 editing in monkey liver, in most cases greater than 60% editing. Keep in mind that about 65 to 70% of the cells in the liver are hepatocytes. Because the LNPs are preferentially taken up by hepatocytes, we believe we edited both copies of the PCSK9 gene in almost all hepatocytes. The editing resulted in substantial reductions in the amount of PCSK9 protein in the blood, as well as substantial reductions in blood LDL cholesterol levels, up to about 60% reductions. Here are the results of ongoing long-term non-human primate studies with PCSK9 LNP base editing therapy. The treated animals are shown in black, and control animals that received phosphate-buffered saline are shown in gray. The results are normalized to baseline levels. The top dotted line indicates 100% of baseline, and the bottom dotted line indicates 10% of baseline, or a 90% reduction. You can see that within 2 weeks, the blood PCSK9 protein level in treated animals dropped by 90%. In following the animals for 8 months so far, we saw that the 90% reduction has been extremely stable, with no sign of attenuation at all. What's notable is that hepatocytes naturally turn over in the liver every 200 to 300 days. So the fact that the therapeutic effect has remained stable over that time period suggests that the effect will remain for the entirety of the animals' lives. Here are similar results for LDL cholesterol. Here, the top dotted line indicates 100% of baseline, and the bottom dotted line indicates 40% of baseline, or a 60% reduction. Within 2 weeks, the LDL level dropped by 60%. Once again, we saw that the therapeutic effect was extremely stable over the next 8 months. Of note, a 60% reduction in LDL matches or surpasses the effects of all of the existing cholesterol-lowering drugs. So if the therapeutic effects of the genome editing persist for the lifetime, as expected, it would be the equivalent of taking a high dose of the most potent statin drug every single day for the rest of one's life, but with a single treatment. Reassuringly, when we assessed for off-target editing with the PCSK9 LNP base editing therapy on primary human hepatocytes, we observed no evidence of editing anywhere except the PCSK9 gene itself. Verve Therapeutics has announced that they hope to start first-in-human clinical trials next year. If the therapy is proven to be effective and exquisitely safe in patients with familial hypercholesterolemia, one could contemplate the use of a genome editing therapy for any patient after they've suffered a heart attack, and eventually for primary prevention. It really could be akin to a vaccination, a once-and-done treatment offered to any young adult who wants it, protecting against cardiovascular disease, the #1 cause of death worldwide, for the rest of a lifetime. If you'd like to learn more about genome editing, or really learn how to do genome editing, or have your trainees learn how to do genome editing, with hands-on exercises and worked examples, I refer you to this book that I wrote that was published just a couple months ago, Genome Editing, A Practical Guide to Research and Clinical Applications. Thanks so much for your attention.